our next presentation by Dr. Jaya Krishnan. He's the CTO of Genome Biologics, experienced senior lecturer with a demonstrated history of working in the research industry, skilled in research, cell culture, scientific writing, microscopy. Professor Jaya Krishnan has more than 20 years of experience in preclinical models and cardiovascular disease research. He completed his PhD at Imperial College in London, UK, and led research at the ETH Zurich Medical Research Council, UK as well, and Imperial College in London. He currently leads an RNA therapeutics group based in Germany, and he has lots to say in his presentation. So now the floor is yours, Professor.
Perfect. Thank you, Sam. Is my screen in full screen? Is that okay? Fantastic. All right. Thank you, Sam, and thank you very much for the organizers for the opportunity to share our work. So as mentioned, I'm Jaya Krishnan, CTO of Genome Biologics, and our focus is to accelerate and de-risk cardiovascular drug development. So I'll just give you a bit of background to who we are and where we are from. So we see ourselves as leaders in RNAi and gene therapy for heart disease. And what we have been able to do is, with the help of EIC support, we have been able to establish an integrated and seamless workflow in order to de-risk.
Sorry to interrupt, Doctor. It looks like your slides are not appearing now on.
Okay. Sorry about this, but we can't go into full screen. So as I was saying, a bit of background about who we are. With EIC support, we have been able to establish this seamlessly integrated workflow, which couples a bioinformatics component all the way right up to biological validation in human tissue mimics. And we are a spin-off of Goethe University Frankfurt, and we have also been residents of J Labs, and we have won a number of grants from the EU, and we are also members of various consortium, one of which is also the Blue Knight Consortium, which is an effort between J&J and the BARDA in the US to increase vigilance for future pandemics. So as part of this, we also have a COVID project where we are trying to identify useful therapeutics there. So now what we are in the process of doing is raising our Series A funding. And with that, I'll just go on to the next slide. Give me a second. Okay, give me a second. Okay, so what's unique about us is the fact that we have this integrated platform which enables us to streamline compounds or targets that we bring forward to the whole development phase. And we do our whole pre-screening platform bioinformatically using a GeniMaps component. This is an AI-driven bioinformatics platform, which we then couple to novel genomics technologies in Genesis and our TrueCardian platform, which are essentially human heart mimics. So what this has been able to do, what this has helped us to do, is to increase the throughput of our pipeline, and the stringency of the assays we perform has also led to the increase in the in vivo efficacy of the drugs that we have moved forward. And overall, this then enables us to de-risk our assets, leading to cost and time savings. So let me just move on to the next slide. Fantastic. So the reason we do all this is that there are huge challenges in cardiovascular disease. The main problem being the risk and cost associated with rare disease and cardiovascular drug development. So unlike other indications, the problem with cardiovascular disease is that of late there have been frequent late-stage failures during clinical trials. So this has increased hesitance and due to it being seen as a more risky endeavor, because this then leads to a decrease in the average in the average ROI. And in cardiovascular disease, there is currently a lack of precision modeling. And this is what we have tried to sort of integrate into our whole screening platform using using the various components of our platform. And so by doing this, we are able to decrease the riskiness and the precision of cardiovascular drug development. So a bit of numbers, some numbers about the likelihood of success of drugs that enter clinical trials. So as it stands, a large fraction of drugs actually drop out from clinical trials during the phase 2 and phase 3 stage of study. It is at this point that the efficacy of the drug is tested either in a smaller patient population or in a larger patient population. And efficacy is one of the biggest hurdles present in order to get a drug approved, and that makes sense. If you identify a drug, you want something that works well, much better than what is currently available. And due to this, clinical trials, especially in CVD, they are long and expensive, and with an eventual approval rate of only about 15%. And so this again then leads to the whole question of how can we increase the efficiency of this process. Now at the start I mentioned that we had this number of platforms and how this enabled us to de-risk our whole development efforts, and this is in part reflected here. So using our platform, we asked the question, now if we think we are able to de-risk drug development, then using our platform, would we be able to say that a number of drugs that enter clinical trials and failed at phase 2 and phase 3 would in fact be detected in our platform as being negative and shouldn't be brought forward? And that was indeed the case. So by doing a blinded retrospective study of a number of drugs that had failed at phase 3, failed at phase 2, and a smaller subset that were approved and went to market, what our platform was able to do was to eliminate 96% of false positives. But the platform correctly identified all drugs that were approved and went to market. So this gives us a degree of confidence that we are able to very stringently remove non-efficacious drugs prior to bringing it forward. So now going into our platform per se. So our platform comprises of three main components, let's say. The first part is the whole optimized disease modeling component. So we utilize big data, DNA-seq, RNA-seq, and single-cell RNA-seq data, both from public resources and also proprietary resources from our collaborators. And what we do is that when we add this data to our AI platform, we are able to very efficiently clean up the data and to then make connections and to identify disease signatures. So one of the biggest problems we see today in this field is the lack of usable data. So there's a lot of data out there, but a large fraction, especially from the earlier years, are not well annotated, and it's not clear exactly what was done in the course of the experiment. Now, being a relatively small company, what we have done is that we have focused our efforts entirely in the human CVD space. And what this has enabled us to do is to specialize and to create a very clean data set of human cardiovascular relevant data sets in order to feed into our system. And these data sets are pre-cleared to only have relevant and biologically meaningful data in our platform. So the output of this platform is essentially disease signatures. What we are able to do is by taking data from different institutions, meaning performing a sort of meta-analysis of data coming from various sources related to different cardiovascular indications, we are then able to identify disease signatures. Now, these disease signatures can be seen as key regulators or key drivers of the whole pathologic process. So once we identify these key drivers, this then enables us to do two things. On the one hand, we are able to then ask the question, which drugs could be efficacious for for a particular indication if we also have the signature profile of these drugs. So then what we do is we essentially look for an inverse relationship between the drug and an indication signature, which enables us to then perform the biological validations to confirm our hypothesis that this drug could be useful for this specific indication. This approach we tend to do in partnership with other entities. In-house, what we do is we use these disease signatures to identify key RNA targets which can be modulated in order to resolve the whole disease situation. So coming from these signatures, we then have developed RNAi-based therapies to determine if their inactivation could actually attenuate disease. Now, the second part of our platform is Genesis. This is a genomics component, which we then can use to model the signatures we identify through our GenMath platform. I will go through this in greater detail in the subsequent slides. So just to go back to the whole GenMath component, as mentioned, we collect big data and we do text mining, both manual and natural language processing based. And then we are able to cluster these data sets according to the indications and further filter out and identify these specific signatures. And the beauty, I think, of our platform is that as we are very focused on cardiovascular data sets, we are able to almost de-skew the data from the vast majority of cancer data sets that are present in public in public databases. And this is an important component because cancer, unlike heart disease, is quite dissimilar. In cancer, you have drivers to increase proliferation, leading to growth of a tumor. But in heart disease, very often what happens in a disease state is that you have a suppression of the ability of the cells to proliferate. And this is actually what leads to heart failure and the death that comes about from heart disease. So lots of the signature patterns are very different. And thus, if you have a database where you have massive bias of cancer data sets, this makes it very difficult to identify the true cardiac disease-related signatures. And this is what we have overcome with our GenMath platform. So from here, as mentioned, we have this technology that's patented in the US, and we expect it to be granted in the EU and other entities soon. And this is our genomics platform. Now, with the signatures that we identify, we are then able to actually implement these signatures in a small animal, a large animal, or in an organoid model, which I will come to shortly. And what this enables us to do is to actually come to a stage of precision, where right now we are looking at population-based signatures. So meaning we are taking a number of patients, categorizing them into population subsets, and identifying signatures specific to these patients. But moving forward with this technology, what we can do is we can start going towards true personalized medicine, where we identify signatures from individual patients and then are able to model them in any of these systems in order to identify most effective drugs for them. And this then brings us to the integration of the two components. So we use our GenMap system to identify targets and to identify drugs with partners. And we are also able to then model these signatures in animals and in organoids to then use as a basis for drug validation and efficacy testing. And this then brings us to our drug screening platform. So I alluded a bit to our cardiac organoids, which is called TrueCardium. Now, this is an advanced human tissue platform, and as I showed in some of the retrospective data earlier, it seems to have high accuracy for identifying efficacious drugs. And the reason why we think it is able to do this as part of this integrated workflow is the fact that, unlike organoids available on the market today, our organoids contain all cell types of the heart, meaning it does not only contain the major populations such as cardiomyocytes, endothelial cells, and fibroblasts, but more importantly, it contains populations such as the neurons and also the smooth muscle cells and parasites, which envelope and regulate the stability of the endothelial cells. Now, for a long time, the neuronal component of the heart has been largely ignored, but it cannot be denied that this is an extremely critical component in the regulation of cardiac contractility, etc. So, by having these cell types in our organoids, enables us to test the effect of the drug not just on the cardiomyocyte component, but rather to get a better holistic view on the action of the drug in all cell types of the organoid, and this is important because if you have a drug which works phenomenally well in a cardiomyocyte, but for some reason kills neuronal cells, this drug is largely ineffective. But of course, in a preclinical screen, if you only had cardiomyocytes, you would not see the negative effects on the neurons or the other cell types. And this is what our platform enables us to see, and is what we think leads to our superior ability to filter out false positives. So what I should also mention is that these organoids are entirely self-organized. So we start with the fibroblasts of a patient, we reprogram them into iPSCs, induced pluripotent cells, and from these iPSCs coming from one patient, we are able to then develop this whole plethora of cell types and the organization that you would see in a native heart. So from this one iPSC, through addition of waves of growth factor and certain mechanical conditions, we are then able to generate all of these cell types and, more importantly, the 3D vascular network of the blood vessels and also of the three layers of the heart: the epicardium, the myocardium, the endocardium, and the lumen right in the middle. So essentially what we have is a mini human heart on a dish. And just to expand on this point further, when you look at this slice of tissue, this is a slice of tissue taken from the organoid by confocal microscopy. What you then see is that the shape of the cells very closely mimic that of the human reference heart tissue, and there's also polarization of the cells, and this is very important for force transduction. Now, these pictures also demonstrate that our organoids represent mature adult human heart tissue, and this is an important point because most of the organoids available today represent that of a early postnatal heart, so not very useful when you are looking for drugs that affect adult human hearts. And adult human hearts and young hearts are very different entities. The young heart has the ability to regenerate, whilst the old heart or the adult human heart does not. And this is just one of the differences. There are metabolic differences, signaling differences, force transduction differences, contractility differences, etc., etc. The list goes on. And for that reason, we have ensured that our organoid platform mimics that of the human adult heart so that it is most useful as a screening platform for drugs targeted at human adults and adults. And this just gives some staining, some data as to the other cell types that I mentioned in my previous slides. Now, as we generated this platform in order to test drugs, we of course benchmarked it to the CVD panel of drugs. So we asked the question, if we were to look at the effect of a subset of drugs that have extensive human clinical data and supporting animal data on how the drug is expected to impact the human heart, what we find is that we have a 95% consistency in the anticipated response in our human organoids compared to clinical and animal data. So this degree of accuracy tells us that our organoids very closely resemble the phenotypic responses of the human heart. Now, when we put our optimized disease modeling platform together with our drug screening platform, this has enabled us to come up with a number of assets. And by going through, so basically these assets are the output or the culmination of our artificial intelligence driven target filtering, signature identification, coming to the whole modeling with Genesis, and the biological validation downstream is through Cardium. And this has led us to a number of assets which we have derived using some of the models that we have established in our human organoid platform. And I would like to focus on one particular asset today, and this is an asset involved in cardiac regeneration. So, as I mentioned at the start, unlike in the cancer scenario where you want to stop cells from over-proliferating and you want to get them under control, in the heart you have the very opposite problem. The heart cells, the adult heart cell is post-mitotic, meaning it will not divide, meaning if it's injured, it will not regenerate, and instead of having new cardiomyocytes being built, what you have is an increase in fibrosis and inflammation, which then leads to loss of contractile properties of the heart due to the loss of cardiomyocytes, which leads to heart failure and death. So this is a huge unmet market. Many of the therapeutics available today do not actually resolve the problem of cardiomyocyte cell death, rather try to modulate the upstream impact of the upstream impact on the heart or downstream impact, meaning the drugs available today target the symptoms and not the underlying cause of the disease itself. And as you see here, it's a huge market both in the EU and in the US, with a total size of about 18.4 billion. So we have tried to address this unmet need using our platform. And in doing so, what we have identified is Gen 115C. So we have identified a number of therapeutics, but today I would like to focus on Gen 115C. So as I mentioned, unlike standard of care, which addresses the disease symptoms but does not actually address the underlying problem itself, with our approach, we have been able to resolve the root cause of the disease. So in myocardial infarction, what essentially happens is that due to an occlusion of a large vessel, this vessel becomes blocked. And as it becomes blocked, then all of the tissue downstream of it, where blood would normally flow, no longer flows. This then leads to an ischemic situation where there is a severe lack of oxygen and also a severe lack of nutrients, because this is one of the key roles of the blood vessel in the transport of oxygen and nutrients. So with this occlusion, the lack of oxygen and nutrients in this area leads to cardiomyocyte cell death. Cardiomyocytes or heart cells are energetically active cell types, and they are particularly sensitive to a loss of oxygen and nutrients. So they die first. When they die, they are then replaced by fibroblasts. Fibroblasts are stromal cells, which play key roles in the maintenance of tissue structure, etc. But they do not confer contractile activity. This is something which is specialized and a unique function of cardiomyocytes itself. So when an infarction happens and you have replacement by fibroblasts and maybe some inflammatory cells, what actually happens is that with time, the heart goes into failure, in the sense that its contractile ability goes down, and if left untreated, it then leads to organismal death. And this we have tried to prevent by inducing the proliferation of cardiomyocytes, of the adult cardiomyocytes themselves. So this is nothing trivial and has been worked on for the past 20 odd years. The problem is that there are many barriers to the proliferation of adult cardiomyocytes. Cardiomyocytes are the largest cell in the body. They are structurally very rigid. So in order to have effective cell proliferation, you need to actually break down the inner cell components to actually have them form daughter cells. And this we have been able to do by using our 115C therapy, where we drive cardiac cell division. So this is a first-in-class RNAi therapy. It overcomes all of the barriers to cell cycle entry, not just the induction of proliferative drivers, but the dissolution of the cells in order to have it plastic enough to actually form two daughter cells. And due to the targets involved in this therapy, which are cardiac enriched, this gives us some form of tissue and disease-specific therapeutic targeting. So I see I'm running out of time a bit, so let me just go into our animal data. So of course we have validated the effect of Gen 115C in 2D and in our true cardiac organoid cultures, and it proved extremely effective, and actually it was used as our screening platform to identify this specific asset. When you go into animals, we can induce myocardial infarction by blocking one of the large blood vessels of the heart. And what you can see is that if you look at the placebo controls, the blue line, heart function very quickly declines, and these animals then die at a significantly higher rate. However, when you induce myocardial infarction in mice but apply Gen 115C, what you then see is a maintenance of survival, and this is due to an increase in cardiomyocyte proliferation, in adult cardiomyocyte proliferation, as you see here. And this then extends to a reduction in fibrosis, because now that you maintain the cardiomyocyte population, you no longer have them replaced by fibroblasts and other inflammatory cell types. And this also leads to a reduction in hypertrophy, which in an overview gives you a heart which is more similar to the control hearts, which are not induced for myocardial infarction. So with that, I would like to thank you all. And we are based in Frankfurt, but we also have labs in J Labs in Berza, Belgium. Look forward to your questions. Thank you.
Thank you so much, Professor Jaya, for your very insightful presentation. I will be opening up the floor to any questions or comments or remarks from the audience, so I'll give another minute or so for everyone if they have any to type it in. And as he shared, you can connect with him and his team via email. And I do suggest everyone follow his company on LinkedIn as well, Genome Biologics, because they do post regularly about their company and updates about their group as well. And I know we have Jonathan Ward from his company participating in the panel discussion later today. So definitely tune into that at 3:50 p.m. So yeah, I'll give another minute or so. And I think, Dr. J, I think you guys have worked with J Labs. Is that correct?
Yes.
J Labs. Yeah, that's not too far. There's a J Labs big incubator here in Toronto.
Okay.
Yeah, no, it's good to see that they're a very vast network of researchers.
Yes. We are at the European site in Belgium.
Oh, okay, wonderful. So, yeah, it looks like so far we haven't received any questions, but if we do, we'll forward them over to you, Dr. Jaya. And again, thank you so much on behalf of Precision Global.
Perfect. Thank you very much.
Take care. Bye-bye.